Good evening, class. I'm Elijah Simmons. Today I'm presenting a paper called Ultra-High Throughout Clinical Proteomics Reveals Classifiers of COVID-19 Infection, published in Cell Systems in 2020. It's really two papers in one, a methods paper about a new way to test blood, and a discovery paper about what that method found in COVID patients. Here's an overview. First, why the study needed to happen at all. Then some background so the science makes sense. After that, how they actually built and tested their method, how the whole study was designed, what they found, and finally where I think this holds up and where I would push back, and of course, strengths and limitations and what's next. The problem. COVID needed fast answers. Back in early 2020, COVID patients were wildly unpredictable. Some people barely noticed they were sick, and there was no reliable way to tell early on which patient had it or not without symptoms. The proteomics technology that existed to look at this closely was way too slow and expensive to use on the hospital floor. So what was actually needed was something fast and cheap enough to run at scale, but still had to meet clinical lab standards. How we got here? This slide is roughly the timeline in late 2019 where the virus started to spread. In early 2020, hospitals got overwhelmed without knowing a good way to sort mild from severe cases. And this paper is basically looking at that gap. Here are some background terms reading blood proteins at scale. And some of the tools that they used. Tools that they have used, one is mass spectrometry, which is basically a machine that can look at hundreds of proteins in a blood sample at once, instead of testing for them one at a time like normal blood panel. DIA or swath MS is a scanning method they use, and liquid chromatography is a step that separates proteins before they hit the mass spec. Normally, getting good separation takes a while, but this paper did it in about 5 minutes. Old way versus new way of scanning. This diagram shows the difference between the old and new scanning approaches. Older methods called DDA kind of random sample which proteins to look at on any given run. So results can vary run to run, like the scatter dots on the left. This The method this paper used, DIA or swath MS, systemically scans everything in a given mass range every time. Shown by the even grid on the right, that's what makes results consistent enough to compare across hundreds of patients. The speed trade-off and how they solved it. Normally, getting good separation quality in the chromatography step takes 20 to 60 minutes per sample on average. This study got that down to about 5 minutes. I thought faster time could ruin the quality and mess with the results. However, side by side against the slow, highly tuned reference method, and they got comparable peak separation quality, so the speed gain didn't cost them accuracy. What we already suspected. Even before this paper, three biological systems were already linked to severe COVID. The coagulation system, severe patients kept showing up with dangerous blood clotting problems. The complement system, which is part of the immune system's first response. But when it overreacts, it ends up damaging the patient's own tissue. And interleukin 6 or IL-6 signaling, that's basically the chemical alarm. The goals of the study. This paper had three main goals. First, build a proteomics platform that could actually survive contact with a real hospital lab, not just sit in a research setting. Second, validate that the faster version doesn't sacrifice accuracy just to save time. And third is to apply it, to run it on real COVID patients. How the study was put together. They started with a patient cohort at a hospital in Berlin, ran their blood through the new platform, did a statistical analysis comparing severity groups, and ended up with 27 proteins that were reliable to track with how sick someone got. Here's our workflow diagram, methods from blood to draw biomarkers. A, so first was a blood sample, goes to a serum or plasma, goes through a standardized automated prep step then 5-minute liquid chromatography separation, then the swath MS mass spec step that reads every protein, then a data analysis stage comparing results by severity. The results are 27 biomarkers, up to 180 patient samples a day, without cutting corners on precision. How they made it faster. 180 proteomes analyzed per day, a chromatography run that now takes about 5 minutes instead of the average half hour, which still reaching clinical lab standard. My natural pushback is I thought making it quicker, cutting the runtime this much, hurts the results. The research for this paper says no, because they benchmarked it against a slower method and got basically the same quality. This is just a visual of that validation point. The two bars, fast method and reference method, are almost the same length, which represents comparable separation quality. You'd see that they're about the same and 1/5 of the time. Who was studied? For the actual patient data, blood came from COVID patients admittedly early to the hospital in Berlin, and everyone was sorted into a WHO severity grade, which is a standard scale. We also ran extra reference samples just to make sure the platform itself was behaving correctly. For analysis, they compared protein levels across those severity groups using differential expression analysis, and they put all their code and lab protocols online so other people can check their work. 27 biomarkers. So the main finding, they found 27 proteins were found to be differential expressed across COVID severity grades. A few of those weren't even thought to be linked to COVID until this paper. What kind of proteins showed up? The 27 proteins broke down into four categories. Complement factors, immune proteins that, when they overreact, damage the patient's own tissue. Coagulation system proteins, the ones behind the clotting issues doctors kept seeing in severe cases. Inflammation modulators, proteins that turn the immune system's response up or down, and the IL-6 pathway, both upstream and downstream signals. Mild versus severe, what's biologically different? In milder cases, coagulation markers stay closer to normal. Complement activity is more contained, and IL-6 signaling stays relatively quiet. In severe cases, clotting-related proteins climb noticeably. Complement activity ramps up and starts damaging tissue, and IL-6-driven inflammation spikes, which is a cytokine storm. What this paper gets right. On the strength side, this is a jump in speed, precision, and cost compared to standard proteomics. It passes regulated clinical lab basic standards. The proteins they found aren't random, they cluster around pathways that already made biological sense, and they open source the whole pipeline so other labs can check their work. Where I would push back on the limitations, one is just one hospital and it's a small early cohort in Berlin. So this needs to be repeated elsewhere before we can fully trust it fully. Correlation, of course, isn't causation. These proteins might just be there synergistically rather than actually driving severity. None of these pathways are exclusive to COVID. Clotting and inflammation show up in planning of severe illnesses, and it came out as a rushed preprint before going through full peer review. Where this could go next, looking forward, I would want to see larger multi-studies to check if the same 27 proteins hold up elsewhere, and it's worth digging into whether any of these proteins are actually good drug targets, not just markers of how sick someone is. So the takeaway, COVID patients in early 2020 were unpredictable, and doctors had no fast way to tell who was going to stay mild and who was going to get seriously sick. So they ran a fast platform on blood from COVID patients at different severity levels, and out of that came 27 proteins that reliably tracked how sick someone was. Also, some of these proteins had never even been connected to COVID. This paper represents mass spectrometry, proteomics, moving from being slow, research-only tool into something that could actually respond in real time to public health emergency. The combination of the two papers is why this paper is useful. Well, that is my presentation, and thank you guys for listening.